Hi, Assalamualaikum and good days to all of you. Today's article about genome characterization and taxonomy of Actinomyces acetigene species novel and Actinomyces stomatis species novel, previously isolated from the human oral cavity. Actinomyces is a gram-positive anaerobic to facultative anaerobic rod-shaped bacteria. It is also known for their rod-shaped morphology and filamentous growth pattern. Actinomyces can form a biofilm, which is crucial in oral and dental disease. Actinomyces is a primary colonizer in oral cavity during polymicrobial biofilm formation. The notable Actinomyces species include Actinomyces neslandi, Actinomyces israeli, Actinomyces odontolithecus, and Actinomyces aureus. Actinomyces is a major contributor to the oral cavity microbial community and various oral disease. Actinomyces understanding advancement. It also include the phenotyping, molecular diagnostic, metagenomics, and single-cell sequencing, has improved Actinomyces species identification and taxonomical. Currently, there are 33 Actinomyces species with valid names. Strain ATCC 49340T, initially identified as Actinomyces neslandi serotype 3. It was reclassified as Actinomyces aureus. Phylogenetic analysis revealed greater diversity within Actinomyces aureus, with strain CCUG 342860 forming a discrete cluster. Actinomyces strain PK6060 overview. It was initially identified as Actinomyces neslandi, but it was preserved and available in the culture collection under ATCC 51655T and CCUG 33930T. It was used as a reference in Streptococcus actinomyces species coagulation studies and focused on the oral biofilm sphere. This study highlighted that an oral Actinomyces revealed that strain from ATCC 49340T and ATCC 51655T represent distinct Actinomyces lineage, leading to the proposal of Actinomyces acetigenes species novel and Actinomyces stomatis species novel, providing insight into oral bacterial diversity. The objective of this article is to utilize the whole genome sequencing and comparative genomic analysis to classify the strain ATCC 49340T and ATCC 51655T as a novel species. Second, analyze the pathogenic nature of this strain and their associate with human infection. The third objective is to conduct CRISPR-Cas9 analysis to reveal their adaptation, immune system, and potential role in defending against foreign genetic element.
Now, I will explain the methodology along this line. For the sample collection and preparation, along with the genomic DNA extraction, the strains ATCC 49340 and ATCC 51655 were utilized in this study. To amplify the 16S ribosomal RNA gene sequence, they employed gene amplification and Sanger sequencing. PCR amplification and Sanger sequencing were conducted using universal primers 27F and 1492R. Then, they analyzed them using EasyBioClad library construction and genome sequencing. High-quality genomic DNA was utilized for library construction using Covaris S2 for fragmentation. The resulting fragmented DNA was quantified using cubic fluorometer. Next, they assessed the quality of the DNA using Agilent Bioanalyzer 2100. The final library was sequenced on Illumina HiSeq X10 platform. Lots of software being used for data processing, genome assembly, and annotation. PrintSec were used to obtain high-quality sequences for subsequent analysis. Quality assessment tool for genome assemblies were used for genome assembly. Genome completeness assessment was conducted through CheckM. RAS were then used for genome annotation, while gvolit2 also used for accurate genome assembly. Multiple approaches were employed to infer the Actinomyces phylogeny. To determine the preliminary identities of the strains, the 16S ribosomal RNA gene sequences were analyzed using the 16S-based identification tool and pairwise nucleotide sequence alignment tool provided by EasyBioCloud. Pangenome sequence analysis pipeline was used for the extraction and identification of the core genome regions and SNPs from each genome sequence. The maximum likelihood tree inferred was based on Kimura 2-parameter model with 1000 bootstraps replicates. Next, the overall genome relatedness index, or GRI, was employed to evaluate the genome of strains ATCC 49340 and ATCC 51655 with available Actinomyces type and non-type strain genomes using the average nucleotide identifier ANI, average amino acid identity AAI, and digital DNA hybridization DDH. Next, the pangenome analysis was held to describe the complete gene set of the most closely related species, including the core genome and unique genome. Genomic islands are regions of the bacterial genome, usually the chromosome, that appear to have been acquired through horizontal gene transfer. In this study, Actinomyces aureus CCUG34288 was used for comparison, while Actinomyces aureus strain T14V was selected as the reference genome to align the sequence genome against. Predicting virulence factors helps in understanding the mechanism of pathogenesis, identifying potential drug targets, and developing vaccines. Understanding pathogens at a molecular level is crucial for developing effective diagnostic tools, treatments, and prevention strategies. This CRISPR-Cas analysis can provide insights into bacterial and viral evolution, antimicrobial resistance, and the development of CRISPR-based tools for genome editing and gene regulation. The protologal description serves as the primary reference for the taxonomic classification of the species and is essential for accurate identification and comparison with other organisms.
Hello everyone. Now I'm going to present the results on the genome characterization and taxonomy of Actinomyces acetigenes species novel and Actinomyces stomatis species novel from the human oral cavity. We sequenced the genome using Illumina iSequence X10, yielding 1469 mpp of raw data each. After filtering, we obtained 1173 mbp of clean data per strain. Genome annotation identified over 3200 genes and 2900 coding sequences in one strand and around 2700 in the other. Both strands showed coding sequence related to amino acids, protein metabolism, and virulence. Phylogenetically, both strands exhibited high similarity with Actinomyces aureus CCUG34288T, but showed genetic distance from other Actinomyces species. Further analysis confirmed close relationship with Actinomyces. Comparative analysis revealed a close relationship with Actinomyces aureus, but the DDH and ANI/AAI values were below the species threshold. Pangenome analysis found over 5,700 gene clusters, including unique clusters in each strand. Both strands displayed differences in genomic islands and lacked exotoxin-associated genes found in Actinomyces aureus, indicating different infection mechanisms. Notably, 8TCC49340T had more CRISPR-Cas genes, suggesting strong defense and adaptability. In conclusion, these novel strands demonstrate distinct genomic characteristics and phylogenetic distance from known Actinomyces species, supporting their status as novel species. Now, I will discuss our findings on the genome characterization and taxonomy of Actinomyces acetigenes species novel and Actinomyces stomatis species novel. Whole genome sequencing provided high quality of draft genomes for both strands with 100% completeness and minimal contamination. Phylogenetic analysis showed close relationship with Actinomyces aureus CCUG34288T while also revealing some genetic differences. We found limitation in using 16S rRNA gene analysis, emphasizing the need for multi-gene and core genome SNP analysis. Genomics analysis, including TYGS, ANI, and AAI, classified both strands as novel Actinomyces species. Cooperative genome analysis revealed a pangenome with over 5,700 gene clusters, supporting the novel species classification. Genomic Highland and CRISPR-Cas genes indicate strong defense and adaptability. Despite limitations such as incomplete genome assembly, advancement in genome-based taxonomic classification have improved species classification and the identification of the new species.
To conclude this article, we can say that the whole genome sequencing and genome analysis of classification of Actinomyces ATCC 49340T and ATCC 51655T as a novel species. It also stated that Actinomyces aureus CCUG 34288T was a closest species. The unique gene and the genome island difference validated their status. It also stated that pathogenicity and analysis revealed that there are association with human or oral infection. Cas9 analysis revealed that there are adaptation immune system and potential defense against foreign genetic element. Lastly, this article proposed that the Actinomyces acetigene species novel and Actinomyces stomatis species novel contribute to the Actinomyces diversity as a novel species. Thank you.